Okay, so I'm recording. Let me share my screen. I'm just going to pull up Moodle first, guys, and then just to kind of update everyone where we're at. And then we can talk a little bit about chapter 5. That's the next chapter that we're going to be covering. So first things first, for in-person lab, I guess I'll just kind of go in order from the top of Moodle here. We do have lab today at 3 o'clock for both either logging in via Zoom at 3 o'clock or being in lab in person. And so if we take a look at the schedule, there's no in-person lab for next week. So I just wanted to remind you guys of that and make sure, you have this schedule handy. The following week will be the diffusion and osmosis lab live, okay? And then I'll talk to you guys about assignments for lab assignments on Moodle in a second. But I just wanted to remind you guys, and I guess this I actually have to change, right? Because there is no lab session B. So it's just 3 o'clock. So let me just do that right now so that that's clear. Right, so we're always going to be meeting for in-person lab at 3 o'clock p.m. Unless there's a couple students that have that class conflict, that obviously doesn't apply to you. Okay, guys? Any questions about this? So you guys can see that so we skip next week, and then there's a lab that following week, and then we have 2 more labs. So these are the only in-person labs that we are doing. Any questions, guys?
So when you say skip next week, you mean like there's no lab at all?
No, well, there's no in-person lab, but there's still a lab.
So it's just going to be online.
Yeah, and we'll probably also still meet via Zoom, at least quickly during lab time, just to make sure I know everyone knows what's going on. But yeah, there's no in-person.
Okay.
Okay.
I'm sorry, I couldn't hear you because I had the AirPods on and they didn't work. You said we will have lab today in person or starting next week?
No, we have lab in person today. Just take a look at the schedule. The schedule is accurate. I was just reminding you guys of that. Okay, there's schedule posted. So today there's in-person lab at 3 o'clock. But next week there is no in-person lab. It's the following week. So again, if you see my screen here, you can see the schedule there. So I was just reminding you guys of that schedule. Okay. Other questions, guys?
Professor?
Yeah.
So for like assignment or chapter 6 assignment, we haven't done that yet.
Oh, what is did it say the due date was now?
Yeah, it said that.
Yeah, that's one I missed. I think I told you guys that. I don't know why Moodle does that. I'll fix it right now. Thank you. So annoying. So your chapter 5 assignment. Oh yeah, 1 second. Sorry, I don't know what's going crazy. Chapter 5 assignment, and I'm just kind of updating you guys, is due next week. So let me just double check that the assignment is correct. Yeah. So on Tuesday, the chapter 5 assignment is due, and yes, obviously the chapter 6 isn't due before chapter 5. So sorry about that. I'll fix that. Let me just give it a second. What is this doing? It should go down there. I don't know why it's not doing that. Okay, where is that? Chapter 6 assignment. So that will be due October 6th. It should be, yeah. Oh my God, what is this thing doing? Sorry. Yeah, so chapter 6 is due October 6th, okay, guys? All right, anything else like scheduling wise or anything? I'm gonna still update you guys in terms of kind of where we're at right now and what's upcoming. Go ahead, somebody have a question?
Yeah.
You cut out. I can't hear you. Can you put it in the chat? I can't hear you. I don't know if it's my Wi-Fi or yours.
All right.
Go ahead, just put it in the chat, okay? Sorry, I don't know why it's cutting out. Any other questions, guys?
Yeah, there was a lab assignment Tuesday, but that was for the lab Tuesday class, right? And now today
Yeah, if you were correct, if you're in lab today, then that chemical composition of cells lab is due today. This lab here.
okay, that one we don't have to do it in lab, it's due before lab.
Correct. This is due before lab. This is separate from the in-person lab, which is here. There's an assignment here that says in-person within this week, right? Okay. And also for this chemical composition of cells lab, please watch the video because I explained the assignment in the video. So you don't have to submit the whole PDF. You're just submitting the mini lab report. Okay. So as long as you watch the video and follow my instructions on the video, there should be no questions. So yes, that's due before your lab time. So if you're in lab today, that should have already been submitted. Okay. If you haven't submitted it yet, that doesn't mean don't pay attention now to try to do it. Just submit it after, at some point today before the end of the day. Okay. And then there will be those of you that were in Tuesday lab already did this in-person lab assignment. Okay. So if you're logging on to Zoom for the lab today at 3 o'clock, you're going to want to click on this assignment here. You guys see this? So I'm within this week, right? If you click on the in-person lab assignment, there's a Word document. That's what we're going to be completing for the in-person lab. Okay, so those of you that are in person, I'll have it printed out for you. If you're on Zoom, you need to then have this with you and you're going to be completing this and then submitting that. Okay, so if you're in person, you don't have to submit it via Moodle. You're just handing it to me. But if you're on Zoom, then you're going to be following along. I will make sure my laptop is in front of somebody, but I recorded Tuesday's lab and kind of just showed the tubes in front of the camera. But anyway, this is what we're going to be completing. We're determining what's in these unknown solutions. Questions about that? Okay. Let me go back to where I was. All right, who put
there was a question in the chat box.
Yeah, let me look at that chat. I just realized I forgot to look at that. Yep. Thank you. I don't know what just happened here. One second, let me just go back. Oh, no. No, so the cell structure and function lab is separate. So that's actually due Friday. So I'll remind you of that too. So normally we would have had another lab that we were doing, this week. So anyway, your lab for last week was that chemical composition of cells, which is due for this week, right? And then we're doing the in-person lab. The cell structure and function, all it is just identifying organelles. And it obviously goes along with the subject matter from chapter 4, which is why I want you guys to just do it this week. So this is the assignment. You just have to open this PDF and basically you're labeling a diagram and writing down the function of organelles, okay? That's due by like 11:59 p.m. on Friday. Honestly, if you need until Sunday to do it, it's fine. I'm not gonna look at them before then, and I won't take off points if you need an extra day or two, okay? So does that answer your question, Katia? You wrote that in the chat there, all right? So it's just page 43 and 45, and it's, like I said, it's super simple, super straightforward. It's just identifying organelles, right? So you're completing this here based on the information that's provided in the PDF. You're describing basic function of these organelles and then labeling them on the diagram, okay? If you don't want to write in the PDF and you want to write it in a separate Word document, that's fine too, okay? Because you could just say for the diagram, you can just put the letters. So regardless, this is what you're completing, pages 43, right? 43 to 45, okay? So again, when you guys, we're gonna have a lab practical, not next week, but the week after, I am gonna have diagrams on there and say, identify the organelle, right? I could also ask you kind of about basic function of the organelles as well. Again, we did that in chapter 4, but we're reinforcing that here, okay? So that's due by Friday. Like I said, if you need to Sunday to do it, it's fine. I won't take off points, or I may just even change the due date. So that's all that is, okay? Questions on that, guys? All right. So, right, we're in module 5. Okay. Again, we're going to do this chemical composition of cells in-person lab, right? The assignment for the chemical composition of cells lab should already be submitted and done. And then you have that cell structure and function lab to do. It's not really a lab. It's more like a worksheet kind of thing. Okay. Chapter 5 homework is due on Tuesday. Okay, so chapter 5 is our chapter that we're starting now, right? We just had our exam on chapters 1 through 4. Now we're moving on to chapter 5. Okay, so what I'll do is I'll pull up chapter 5 and talk a little bit about chapter 5, but I want to just make sure we all kind of know what's due in terms of assignments. For the exam, I plan on trying to get through grading the exams like tomorrow, but certainly through the week, by the end of the weekend. Okay, and I don't know if you guys know, there is something called, well, they call them 3-week grades, but they're really not. They're kind of like a third. So instead of just having midterm grades, we have kind of, your grade will be posted through the first third of the semester, and then kind of the second third, and then you'll have your final grades. So those grades are due technically by Friday, but usually she gives us a few extra days. So certainly by Monday, those grades will actually be posted on self-serve. It will be the same grade that you guys see on Moodle. Okay, so you guys should be checking Moodle to see your grade. So basically, I'll have everything graded up until exam 1. So that grade that you see on Moodle, or that grade that I post on, not Blackboard, that I post on self-serve will be accurate. Okay, so it's gonna give you a chance to see where you're at in terms of a grade in the class right now, all right? Anyone that's below a C+, I will put in a care report for, okay? Just so you guys know. So once a care report is just about, hey, you know, your average is this, and basically your advisor gets flagged, and so does Miss Borhill gets flagged, and they'll kind of follow up with you, okay? So if you see that, that's why. Anyone who has a grade below a C+, a C or below, you're gonna get a care report, okay? Questions, guys, about that?
Professor, will that be after the exams or before?
What,
after the exam is graded? Yeah.
It's gonna be after the exam is graded. Otherwise, it's kind of an inaccurate assessment. So yes, after the exam is graded, that grade will be pretty accurate. Yeah, if it doesn't include the exam, then it sort of doesn't really make sense because the exams take up the majority of your grade. Okay, so after the exams are graded, everything else will be graded, the labs that you submitted and everything like that, and all those grades will be inputted. So that grade that you see, and again, I haven't graded it yet, so whatever you see on Moodle is probably not really that accurate right now until all of those grades are inputted in, okay? But that'll be accurate. That's a good, you know, assessment of where you're at in the course, you know, right now, all right?
Okay. Thank you.
Yep, and if you guys have questions about it, then you let me know, right? Again, this is meant to be, you know, transparent, so you guys know what your grade is all the time. And if you have questions, you can ask, okay? But like I said, what you see on Moodle, it will then be posted on self-serve, but it's the same grade. I'm just gonna translate that number grade into, you know, into a letter grade, okay? And the scale is outlined on the syllabus in terms of like what's an A, right? What's an A minus? That's all outlined on the syllabus, okay, guys? So it just goes straight based on your average. Whatever that number is, that's what grade you get. There's not really any question. Any questions about that, guys? Okay, all right, so like I said, we are, in terms of where we're at for assignments, that chemical composition of cell mini lab report, right, is due, okay? When you come to in-person lab, you're gonna be completing this in-person lab assignment, okay? Some people already did that because they were in Tuesday lab. Then remember, you want to complete this cell structure and function assignment, okay? Like I said, it's not really that hard. It's labeling a cell diagram and writing down functions, to which we should already know because we just had an exam on that, right? Okay, and then chapter 5 homework is due on Tuesday, okay? And then moving forward, we'll have another quiz, right? I think I have, yeah, I'll probably schedule a chapter 5 quiz for that following week or whatever. So just kind of be, you know, just pay attention. I'll remind you guys about that. So for right now, we're gonna worry about listening to the chapter 5 recorded lecture, right? Maybe having our review sheet out when we do that so that we can take notes on it. Remember, there is a review sheet for exam 2, and it's posted where the exam is. If we ever have exam 2. Right, so exam 2 is scheduled right now for the week of October 19th, just so you guys know, okay? If that changes, I'll let you know, but right now that's when it's scheduled for, either that Tuesday or Thursday. The exam 2 review sheet is there. So exam 2 is chapters 5, 6, 7, and 8. So, and these are pretty dense chapters, okay? So it's a lot of information. So make sure, again, you're keeping up on it. You should have that exam 2 review sheet out with you every time you're listening to the recorded lectures, and you should be taking those notes. You will then be submitting the picture of the completed review sheet right before that exam. Let me make sure that that date isn't some weird date. No.
I have a question.
Yeah.
Do we necessarily have to use the review sheet? Can we just take notes on our own?
You can, but it's probably to your benefit to look at the review sheet, right? Because those are the points that I think are important, right? But yeah, you're okay if you're just taking notes on your own, that's fine. But make sure, like I said, you kind of have that review sheet next to you so that you're making sure your notes are encompassing what's there. But yeah, okay. But as long as you're taking notes, I guess that's the point, right? And some people, so that's okay if you submit those notes, that's fine. But like I said, make sure you're still looking at that review sheet though. All right, so let me pull that review sheet up. And before we take that second exam, there will be a lab exam, okay? I always give lab practicals. Usually when we're live, you're doing them actually in lab and you move from station to station. There's pictures, there's microscopes. Obviously you can't do it that way, but it is going to be on Moodle, just like everything else. There will be pictures of cells. There'll be, you know, pictures for you to identify things based on the labs that we did. So I'll review for that. So don't, you know, just a heads up, but all of the labs that we've done up until, you know, essentially up until the diffusion lab, that's what's going to be on that first lab exam. All right. So it's not like you guys just do the lab, don't pay attention, and then you're not going to be asked about it. We really have to make sure we understand a lot of these concepts. Okay. So I'll review. There also is a review sheet for the lab exam that's up. Okay. So if you're curious, you can kind of take a look at that, but I wouldn't worry about it right now. All right. But I just wanted to give you guys a heads up. Okay. So for exam 2, right? Chapter 5 is the first chapter we're going to cover on exam 2. Okay. So that's what we want to start on. It's membrane structure and function. Okay. So we'll talk about the different components of the plasma membrane. Or I already talked about this in the recorded lecture, right? So there are, I guess, let me just leave it at that. I'll pull up the figure. I'll pull up chapter 5 and talk specifically about those components. Okay. But one of the components, or glycolipids and glycoproteins, are components of the plasma membrane that maybe we don't usually think of. And all that means is that they have carbohydrate chains attached, right? So a glycolipid is a lipid with a carbohydrate chain. A glycoprotein is a protein with a carbohydrate chain attached. Okay, that's what I meant by this. All right, and then we talk about different functions of membrane proteins. Okay, so let's start talking about some of these concepts. And then chapter 6 is going to focus on a lot on energy in general, but then also enzymes, right? And function of enzymes. And then there's a lab that goes along with this, which is the enzyme lab, but that's a couple weeks away. Okay, so there is a lab that helps demonstrate that as well. And then 7 is photosynthesis, and chapter 8 is cellular respiration. So like this exam is probably a lot harder than the first exam, just so you guys know. So make sure you're staying up on the material. And if you have questions, you know, I can clarify, I can go over things, ask Katie during tutoring, whatever you need to do. Okay. You can't say, if you save it to the last minute, then you're going to put yourself at a disadvantage. So just make sure you're keeping up with everything. All right, guys. So let me pull up that chapter 5 lecture. And let's take a little bit of a closer look on that. So you're listening. So again, we want to make sure we listen to that recorded lecture. We do that chapter 5 homework. And then next week we'll start chapter 6. Okay. That's not what I meant to do, but there's the recorded lecture. In case you guys didn't know, I meant to hit the PowerPoint. Okay. All right, so I guess just to begin, if anyone listened to the lecture, or looked at the PowerPoint. Does anyone have kind of anything that they specifically want me to go over right now, or something that they kind of remembered that was a little confusing or anything like that? All right, so let me wait until that downloads and then I'll pull up that lecture. Apparently my Wi-Fi is slow. All right. It's coming. Okay. All right. So like I said, this entire chapter, right, focuses on the plasma membrane or the cell membrane. Okay. So what is the plasma membrane? We mentioned this in the last chapter when we talked about different organelles, right? What's the plasma membrane and what's the function of it? Just basic function. Somebody tell me. Where is it, I guess, first? Think about the cell. Where's the plasma membrane?
Isn't it in the nucleus? Nucleus? Excuse me. I'm sorry.
So,
Plasma membrane?
the nucleus has a membrane, right? A nuclear membrane. But when we talk about the cell membrane or the plasma membrane, what are we referring to?
Something that surrounds the cell. So,
Go ahead.
yeah, so basically its function is to allow some substances in and out of the cell.
Good. Yeah. So it surrounds the cell, right? And just like Moses said, it lets some substances in and some out. And basically the point is that it regulates what goes into the cell and what goes out of the cell. Okay. And then obviously it's not as simple as that, but let's just kind of think about that, right? It just surrounds the cell. And again, it's going to regulate what goes into and out of the cell. So what are some examples of things that the cell wants to take in or needs to take in, I guess? What do you think?
Water.
Okay, good.
Protein.
What else?
Protein.
Okay.
Protein.
What's used as an energy source for cells?
ATP, mitochondria.
So in the mitochondria, correct, ATP is produced, but from what? What's the molecule that the cell breaks down in order to produce ATP?
Fatty acids. I mean, is it fatty acids?
Well,
Amino acids?
the cell can break down fatty acids to produce ATP. Okay, so sure. But what else? What's the main one that we usually think about? So like if we talk about cellular respiration in the mitochondria, we talk about breakdown of glucose, right? So the cell has to be able to take in glucose, right? Things like that. And so if you guys think about, right, you think about like diabetes, or you think about blood glucose levels, right? Glucose travels around in your blood, or you think about you ate something, right? It gets digested, broken down. That glucose makes its way ultimately into your bloodstream, right? So that it can then get delivered to your body cells. Once the glucose encounters the cell will take that glucose in, right? So you don't want the glucose in your blood, you want it inside your cell, okay? And that's a process that involves the cell membrane. It has to get into the cell. So that's just an example of something that needs to get in. You guys gave other examples as well. And then there's waste products that the cell produces, right? And those would need to be released from the cell, okay? So the take-home message is that there are always things or molecules coming into and out of the cell, or gases, right? Oxygen and carbon dioxide, they have to be able to get into the cell as well, okay? So anyway, that was just kind of us to kind of think about big picture, I guess, okay? So if we talk first about the structure of the plasma membrane, right? I guess we can go through this little example that's here. All right, before I start talking about structure. So we're just talking about the plasma membrane, right? And we're talking about things going into and out of the cell, right? The plasma membrane is made up of a phospholipid bilayer, but there are also many other things in the plasma membrane, okay? One of which is something called a receptor, okay? So have we heard the term receptor before? When you think of a receptor, what do you think of? Anyone?
I think of like a neuron.
Something that picks up signals.
Yeah, like
Okay, good.
receives information.
Okay. Yeah, kind of, yeah. And where do you usually think of receptors being? Or do you think, I mean, like I usually, when I think of a receptor, I think of a drug, right? Or something binding to that receptor that then you're right, will initiate some sort of response. And you can kind of think of it as, I don't know, conveying a signal, I guess.
I think of nerves,
What do you think
of your nerves.
Okay, so why do you think of nerves?
Because nerves are like what send signals all over your body.
Yeah, and I guess if you're thinking of kind of receptors or receiving information, right? You think of your nervous system as being that system that's going to take information in, right? And bring that information to your brain, essentially. If you think about sensory information. Okay, that's all right, that's good. If you guys look at this example here, right? It's referring to a substance called capsaicin, okay? So this is a substance that's in hot peppers, right? Things that are hot that actually make your mouth feel like it's on fire, that feeling of it being hot. And basically how that works is that capsaicin, again, it's a chemical or a molecule that's in the peppers. It will bind to a receptor, right? Or a protein that's in the plasma membrane of cells in your mouth, okay? It will then activate those cells, and that's where you get the pain, the feeling of pain or the feeling of fire kind of thing. So the idea is that the receptor sits on the surface of the cell, right? And then that capsaicin can bind to it. And then once it binds to it, it basically activates that cell, right? To then give you the feeling of something being hot. Whenever you think about, drugs, pharmaceutical drugs or otherwise, right? In terms of how they exhibit actions on the body, they bind to some receptor on a certain cell type. If you're talking about like psychoactive drugs, usually their action is in the brain, right? And that's what's then going to lead to these, the effects that you feel. Okay, so any drug, even any hormone or anything like that in your body, it has to bind to a receptor on its target cell in order for it to exhibit its effects. Okay, and often those receptors sit in the plasma membrane, so that's why we're talking about them now. So the first part of this chapter talks about the structure. Okay, so and again, we probably have already talked about this. Okay, we talked about all this already. We know, hopefully we remember that it's a phospholipid bilayer. Okay, remember we talked about the hydrophilic heads of the phospholipids and the hydrophobic tails. Okay, but there's more to it than just that bilayer. Okay, so we mentioned the bilayer. Okay, but there are also proteins like those receptors that we just mentioned that sit in that phospholipid bilayer. Okay, also cholesterol is a part of the membrane as well. Okay, so if you guys look here, right, it's kind of breaking it down into three main components. There are kind of others that sort of, I guess everything kind of fits it within these three main components, but I'll show you specifically examples of everything that's in the plasma membrane in a second. But there's a lipid component, right? We talked about the phospholipid bilayer, okay? There are also other types of lipids called glycolipids that are part of that membrane as well. And then there are protein molecules that are in the membrane, okay? And these protein molecules can actually move around the membrane. So that's why it says they float around. And if you think about these membrane proteins, they can kind of be of two main categories here, right? Either peripheral proteins that are and I'll show you a picture. They're sort of, they face the inside of the cell and they don't really attach directly to the membrane. And then you have integral proteins like receptors, which basically span the whole membrane, okay? Or the bilayer. Cholesterol, like I said, is part of your plasma membranes, and it will affect the fluidity of the membrane. So it's an important component of the plasma membrane. All right, so if you guys look here, this is showing you some of those components that we just mentioned, right? The phospholipid bilayer, this is showing you cholesterol kind of wedged in here. This is an integral protein. It spans both of the bilayers, or spans the bilayer, both layers. And then peripheral proteins kind of hang off the outside here, facing towards the inside of the cell, okay? Like I said, there's more. Okay, so I kind of alluded to this already and mentioned this, but in addition to those three sort of main components that we mentioned, there are also glycoproteins and glycolipids. Okay, so everything that I just talked about, all of these things are part of the plasma membrane. So like I said, what's a glycoprotein? It's a protein that has a carbohydrate chain on it. Okay, glycolipid is a lipid with a carbohydrate chain, and these have specific functions. Okay, and we'll mention that when we talk about some functions of plasma or of membrane proteins. Okay, all right. You only see these carbohydrate chains sticking out on the outside of the membrane. Okay, so if you look here, see these little yellow things here? These are the carbohydrate chains. So here, this is a protein, right? An integral protein, and has a carbohydrate chain sticking out. That's a glycoprotein. You can see, do I have a glycolipid? Yeah, and then here showing you an example of a glycolipid. Again, these are the carbohydrate chains, and they only stick out to the outside of the cell. Okay, so you don't see carbohydrate chains stick to the inside of the cell. All right, so we should know all of these components of the membrane. Okay, we just went through pretty much all of them, and if I showed you a picture, you should be able to identify what's what, right? We should know that this is an integral protein, whereas this is a peripheral protein. Okay, so the peripheral proteins are not directly attached to the membrane. If you notice, the peripheral proteins are actually attached to another integral protein. Okay, but bottom line is main difference: integral spans the bilayer, peripheral proteins kind of hang on the out or towards the inside of the cell. Does anyone have a question? Anyone have a question so far about the components of the membrane? All right. So then, like I said, I just mentioned, right, that we would talk about functions of different membrane proteins. Okay. So the integral versus peripheral proteins are really just two main categories of proteins. Here now we're talking more specifically about different types, right, and things that have different functions. Somebody wrote something in the chat. One second, let me just check.
Dr. Komeski, it's me.
What's up? You wrote in the chat?
Yeah, I just sent you a private message. I'm currently having an internet connection issue, so I'll be dropping out.
Okay. All right.
Yeah.
Are you at school on campus?
I'm at school, but it looks like I'm on campus, but the Wi-Fi is off.
Sure.
I don't know how, but all of them is off. So.
Okay. All right. Thanks for letting me know. Okay. So if we go back again to talk, now we're going to talk about functions, right? Like I said, so you guys are responsible to know about these different types of membrane proteins that exist and what their functions are, okay? So there are channel proteins. We said one of the main things about the plasma membrane is that it allows things to go into the cell and out of the cell, okay? Some things can actually diffuse across the membrane on their own, but a lot of things have to go through special proteins, right? Called channel proteins or carrier proteins. Okay, so channel proteins, just like the name suggests, right, allows molecules to pass through that membrane via a channel. I'll show you a picture. Carrier proteins, they do the same thing, but they just move things across the membrane in a different way. Okay, so the substance kind of binds to it and then it gets released. Another type of protein, cell recognition proteins. So remember I mentioned those glycoproteins, right? They have the carbohydrate chains that stick out to the outside of the cell. These are, you can think of them as being cell recognition proteins. So one of the examples is, it says here, it helps the body recognize foreign substances. So there are specific kind of carbohydrate chains that sort of stick out on your body's cells that tell your body or tell your immune cells that this is from, you know, this person, this is self. And if something doesn't have that, then the immune cells will consider it as a foreign material and attack it, right? And so these are glycoproteins that do that. Okay, so that's why we call it cell recognition. It's the body's way of identifying their cells, right? The cells, and so that the immune system knows that these are not foreign. These are cells that belong here, essentially. Okay. Questions so far, guys? All right, so we mentioned receptor proteins, right? In the beginning, we were talking about capsaicin binding to its receptor, and that's what essentially initiates that response, that feeling of something being hot. Receptor proteins in general will bind specific molecules. Like I said, whether it's a drug, whether it's a hormone, that drug, that hormone that's traveling around in the bloodstream, it'll reach its target cell and bind to the receptor, and then that will initiate a response, right? So then that cell responds to whatever that hormone or that drug was, okay? So again, a drug or a hormone floating in your blood, right, serves no purpose. It has to find its target cell to exhibit any sort of physiological action, okay? And it does this by binding to receptors. So that's a super important, just main general concept of biology that we should understand. Also, there are enzymes that sit in the plasma membrane, right? Enzymatic proteins that help carry out metabolic reactions. And I told you guys when I was going over that review sheet for the next exam, in chapter 6, we will talk about enzymes a lot, okay? There are also proteins called junction proteins that exist in the membrane, and they help basically attach cells together. So when we think about cells forming tissues, right, they'll be joined together by these special proteins, okay? Now, the next set of slides explains all of these types of membrane proteins in a little more detail, and then also gives you some examples to try to sort of relate, okay? So I'm not gonna read over again what I said, but if you look at a picture of the channel protein, right, it's showing you, it's gonna allow things to move across the membrane, okay? One example here is that, I mean, we've heard of the genetic disorder cystic fibrosis, maybe, probably, because for some reason, I always use that as my example. But basically what goes wrong, or what's wrong in patients with cystic fibrosis, is that they have a chloride channel. So it's a channel like this, and chloride ions normally will move through it, okay? It doesn't work, it's faulty. And that then affects the movement of water and leads to this really thick mucus that will accumulate in the respiratory passages and also in other ducts, pancreatic ducts and things like that, okay? So giving you kind of like a why do you care? Like who cares about a channel protein? Well, if you have a defect in just one single channel protein, it leads to a very serious disease, okay? And so the other thing to mention here is that these channels are specific, meaning you have a specific channel for a chloride channel. You have a specific channel maybe for sodium ions, okay? So these are specific. They're not just general kind of tunnels that anything can move into or out of, okay? Because the cell has to make sure it tightly regulates what gets in and what gets out. All right, so if you move on, like I said, there's gonna basically be this for every single one of those membrane proteins that I mentioned, okay? Carrier protein, again, similar to the channel protein, except that the way it moves moves molecules across is slightly different, okay? This mentions sodium-potassium transport, okay? And that this actually has been connected to obesity. That's sort of a more general kind of statement, but I think, again, making you understand that the movement of these ions, right, across the plasma membrane is really important, and it's important that it's regulated right? That what's supposed to get in, gets in, and what's supposed to get out, gets out, okay? All right, what's our next one? Cell recognition protein, I mentioned this, okay? So again, this is a glycoprotein, right? You see the carbohydrate chain sticking out. These act as identifiers for the cell, okay? So there's one specific complex called an MHC complex. So again, this is the idea that of identifying something as being foreign versus self, okay? So it says here, organ transplants are difficult to achieve, right? Because again, every person has this specific MHC complex. And so they'll get, if you put anything that's foreign, right, a foreign organ in, it can easily get attacked by the immune system, okay? All right, receptor protein, we mentioned this already as well, right? Here's your substance like capsaicin. It has to bind to its receptor and then will initiate some sort of effect on that target cell. Gives you an example here with pygmies, okay, and growth hormone. So it says pygmies are short not because they don't produce enough growth hormone, but because the receptor is faulty, right? So again, it doesn't matter how much of a drug or a hormone is floating around in the blood. If it cannot bind to its receptor on its target cell, it has no biological action, okay? Enzymatic proteins, again, these are enzymes, okay? These sit in the membrane, but they'll catalyze metabolic reactions, okay? So this is giving you an example of an enzyme called adenylate cyclase, okay? It's involved in ATP metabolism. Again, I'm not expecting, or like, I don't want you guys to memorize any of this, but I mean, I guess I have in the past maybe asked for like an example. Like I may give you a choice, right? Like I may say, hey, tell me what these 3 membrane proteins are, right? Tell me the function of them. And then maybe I'll say, give an example of one of them, okay? So I don't expect you to memorize all of these, but you should at least know 1 or 2 examples. So like for the channel proteins, an example was sort of the chloride channel, right? And we gave an example, or I might say sort of give an example of an effect. So like if I said, okay, here's a channel protein, tell me what would happen if somebody had a faulty chloride channel or something like that. Or if a channel protein wasn't working properly, how could that affect the cell, right? It would just be something kind of in general so that I knew you understood that channel protein is responsible for getting things into and out of the cell, okay? So that's kind of along the lines that I would ask sort of a question related to this, okay? I don't expect you to like memorize the function of adenylate cyclase. These are just meant to be examples, okay? All right, junction proteins. I said that these are proteins that exist in the plasma membrane, and they sort of link 2 cells together. So you can see that the proteins from this cell in this plasma membrane are interacting with the ones from this cell. And again, it's going to hold cells together. So it gives you an example of during neural tube development, right? Or during development of the nervous system, that you have kind of like, I don't know, you can kind of think of it as sort of like tissue remodeling. So cells kind of differentiate. I'm trying to think of a simple way of putting this. So you'll have a lot of, if you think about during development, right, just in general, you think about, you have one single cell to start, right? A fertilized egg. And then you have a whole bunch of cell proliferation that happens, right? You get a whole bunch of cells that are produced. Then those cells will become, those stem cells will become specific, right? They might become brain cells, right? Nervous system cells. And then what's going to happen next is you have to form tissues, right? And so the cells need to be able to interact with one another to form tissues and then to form organs. And that's kind of the example that it's giving you here. You can also think about, if you think about like the lining of your GI tract, right? You eat food, goes into your GI tract. What's going to cross through the GI tract and get into your blood is only the digested molecules, right? The cells that line your GI tract are very tightly packed so that things can't just leak in between, okay? So that forms this epithelial tissue that lines your organs. So that's kind of another example as to why these kinds of proteins are important. All right. And that's the last one, okay? So again, make sure, right? I just spent a lot of time going through the different types of membrane proteins and their different functions. You guys need to know that, right? So you should definitely be able to list all of these and tell me what the function is. Again, I don't necessarily expect you to memorize the examples. The examples are just there to kind of give you a why do you care, sort of what does this apply to kind of thing, okay? Are there questions about that, guys? All right, the next part of the lecture, again, I don't want to go through this whole lecture. I already did this lecture. It's already recorded, but I really wanted to make sure I went through that because I do expect you guys to be familiar with all those membrane proteins. The next part of the lecture talks about the permeability of the plasma membrane, right? And kind of what things can get through and what things don't, right? And a term that we use is that the membrane is selectively permeable, okay? Again, some things get through, some things don't, okay? In general, small non-charged molecules, okay, will freely cross the membrane. So things like carbon dioxide, oxygen, glycerol, alcohol, they can diffuse across the membrane on their own, okay? So they don't have to go through a channel or anything like that. They don't bind to a cell surface receptor or anything. They can actually move through the membrane themselves, okay? And one thing that it says here is that they move from high concentration to low concentration. So what do we call that? What's the term to describe when something or a molecule moves from high concentration to low concentration? One word.
Diffusion.
Yeah, thank you. Diffusion. Okay. So when we talk about diffusion, it's the movement of molecules along their concentration gradient, right? From high concentration to low. Okay. So these substances that I mentioned here, you know, if there's a high concentration, say, of carbon dioxide outside the cell and a low concentration inside, the carbon dioxide would just freely diffuse across that membrane and move to the inside, okay? These guys can just diffuse right across on their own. Water, okay, I think that was one of the examples that you guys gave, right? When I said, what does the cell need to take in? And somebody said water. And so that's true, for sure, right? The cell uses water for a whole bunch of stuff. And actually water moves across the membrane in special channels called aquaporins, okay? So water technically could get through the membrane on its own, but the rate that it would be able to do that is irrelevant. And so it moves through these specific channels, or I'm labeling it here as specialized proteins called aquaporins. Okay, so if I ask you, how does water move across the plasma membrane, right? Or what does it move through? The answer is aquaporins, okay? All right, so ions, right? Remember I gave the example of the chloride channel. Chloride's an ion, right? Sodium ions, potassium ions, all of these cannot pass through the membrane on their own. They have to move through channels, okay? So this is all passive transport or diffusion, right? We're talking about things moving along their concentration gradient. Now I have mentioned here something called active transport. So what does that mean? When we say it's active, what does that suggest?
Movement.
Okay. And what do you think? If something's moving against its concentration gradient, meaning, right, it's moving from an area where you have low concentration to high. So it's moving against the way it would normally have a tendency to go. So what do you think? Do you think that would require more effort? Requires energy, right? I think I was
more energy is
yeah.
required.
When you say when you say active transport, it's going to require energy. And that's what this says here. So you're moving against the concentration gradient. So I don't know, you can think about like, if you're walking your dog, right? I mean, I guess if you have a little dog, it's easier, but if you have a big dog like I have, right? And you're walking him and he wants to go this way. All right, it's fine. He's walking along, doesn't really require much energy, right? But if I'm trying to pull him against the way he wants to go, it's going to require a lot of energy, right? So you're moving molecules against their sort of tendency to move, okay? And so it requires energy. We call that active transport. There's also something called bulk transport, where the cell can take in larger particles, okay? So actually a cell can engulf a bacteria, okay? That's called endocytosis. So endocytosis is bringing something in, exocytosis is releasing something. So these are larger particles. Okay, so these were all examples of movement of things across the membrane. Okay, so here's an illustration of that. Okay, so here's your cell, here's inside, here's outside. Remember that there is a very high concentration of water both outside the cell and inside the cell, right? And that dictates how this bilayer sets up, right? Because what about these phospholipid heads? Are they hydrophilic or hydrophobic? What did I just say? Hydrophilic, right? These guys are hydrophilic. like water, they face towards the water. And then what about the tails? What are they?
Hydrophobic.
Yeah, hydrophobic. And that's why they face towards the inside, because they're trying to get away from the water, okay? All right, so now if we kind of look at what's gonna, kind of some things that might cross, right? This is showing you here. Charged molecules and ions, they can't get through the membrane on their own, right? They have to move through channels. That's what this is showing you, okay? Let's see what we got next here. Water, how does it move through? Moves through aquaporins, its own specific channel there, okay? Now remember, these ions do have channels that they can move through, but this is illustrating the fact that they can't diffuse across the membrane on their own. They have to go through their channels, okay? Non-charged molecules, like small non-charged molecules in general can cross, okay? Large molecules like glucose can't cross, it can't cross the membrane on its own. It has to have help, okay? All right, so these are some terms that I mentioned already, okay? And we should be familiar with this. So again, if we talk about diffusion, all diffusion is, and we'll go over this, we're gonna have a whole lab on diffusion, we'll go over this multiple times, but diffusion is just movement of molecules along their concentration gradient, right? They move from high concentration to low, okay? It says here examples. So this is saying, okay, what are some examples of molecules that can diffuse across the membrane on their own? Okay, so lipid soluble molecules and gases, carbon dioxide, oxygen, they can readily move across that membrane. If you talk about facilitated transport, these are molecules that have to move across the membrane through a channel, right, or a carrier protein. Okay, so some sugars, amino acids, ions, right? Those have to move through channels, okay? Active transport, so it says here, so now in this case, active transport, you're moving the molecule against its concentration gradient. So it says here, a carrier protein plus energy. So it requires energy, and often we call these pumps. So you're pumping something against its gradient, okay? And so I don't really, this is kind of, so I guess it's a little misleading. So ions can move against their concentration gradient, but there's also examples where they move along their concentration gradient. So you can kind of have ions in both categories here, okay? Same thing here, right? You can see amino acids are in both categories. Sugars are in both categories, okay? So it only becomes active if it's moving against its concentration gradient. But the bottom line is all of these molecules have to move through channels, some sort of channel, because they can't get across the membrane on their own, okay? And then if you talk about bulk transport, right, macromolecules, even a whole bacterial cell, that would be engulfed, right? And we call that bulk transport. But again, you can also release things, which would be exocytosis. So that's the other way, but bulk transport is large particles, okay? What's a vesicle? Remember we mentioned vesicles in the last chapter? Why does it say vesicle utilization? What the heck does that mean?
Isn't that like a little, like a bubble or a pod or something that they could tap in?
That's kind of a good way of describing, I guess, right? It's kind of like a little bubble, like a membrane, right? We talked about that endomembrane system, right? Like if you think about like a protein being made in the cell, right? Remember? And then sometimes the proteins have to be secreted from the cell. And the way that they get secreted, they get packaged in a vesicle, right? And then released. So that's what that's referring to. And then if you think about something coming in, when it comes into the cell, some big thing like a bacterial cell, it'll also come in sort of as a vesicle and then get transported, you know, to the lysosome or whatever to be broken down, okay? So yeah, that's a good description, okay? All right. Okay. So now the next section focuses on, you know, really illustrating the concept of diffusion, okay? And again, the movement of particles from high concentration to low, okay? So a couple, you know, something that you kind of need to know right off the bat, I guess, if we're thinking about solutions, right? And in a solution, you have a solvent, which is a liquid. In our case, in biology, and we're talking about cells, that solvent is usually what, guys? What do you think that solvent is if we're talking about like a cell?
Water.
Water. Thank you. Okay. And then solute is some sort of dissolved solid. So I know we can think about like sodium chloride or something like that. Anyway, there's some sort of dissolved solid, and there are many examples, I guess, we can think of in biology in terms of a solute. You can even think about glucose as being a solute, right? But water is most always the solvent. Okay. So let's look at a picture of this, right? And we want to understand what's written here, but let me do that by showing you a picture. So this is like a super easy illustration of diffusion. Okay. I don't think the fish were harmed by the dye molecule that was in there, just so you guys know. So here's your fish tank. Here's your, just showing you like one little speck, like piece of a dye, right? A crystal or whatever. What's going to start to happen? Okay, you got a high concentration of solute or crystals, dye crystals here, and a really low concentration everywhere else, right? So if we talk about diffusion, what's going to start to happen, guys? What's my next picture going to show?
You're going to see the dye start to disperse in the water.
Yeah, good. You're going to see the dye start to spread out, right, in the water where there's not a lot of dye molecules, right? Moving from high concentration towards low, okay? So when we're thinking about diffusion here, right now we're describing the movement of the solute, right? The dye in this case, okay? So let's go next. So that's exactly what's happening, right? Oops. You can see here, right, your diffusion of these dye molecules kind of extending out into the water. And then obviously over time, right, you're going to get an equal distribution of dye molecules and water, okay? And so we call this equilibrium. One thing to remember or to think about is that even when this reaches equilibrium, there's still movement, okay, of the molecules. But the idea is there is no more net movement of this molecule in any one specific direction, right? Here, the net movement is outward, right, where the water is. But now once you reach equilibrium, there is no more net movement, okay, in one specific direction, okay? So we say you got equal distribution, it's reached equilibrium. And on that last slide, I think I have that term there. Let me just make sure, right here. It says when net movement stops, okay? All right, now, one thing to kind of think about and to put this, I guess, in a biological sort of perspective, right? That's a dye molecule in a fish tank. And by the way, you probably shouldn't do that to your fish. They'll probably die. Though those guys looked okay, but like I said, they'll probably won't make it. If you think about gas exchange in the lungs, so what did I tell you? I told you that carbon dioxide and oxygen, right? They readily diffuse across the membrane, okay? The movement of oxygen, this is particularly focusing on oxygen. So let's just focus on oxygen. I don't think I show carbon dioxide, so we won't even go there, okay? Yeah, we'll just focus on oxygen, okay? So let's think about this. You inhale, right? You take air into your lungs, right? That air ends up going into these tiny little air sacs in your lungs called the alveoli, okay? If you want to say it in one single alveolus, okay? And so when you're breathing in, you're hoping to get oxygen, right? And so you got a whole bunch of oxygen in there in this little air sac here. And what surrounds them is blood vessels, right? A capillary network. Because where do you want the oxygen to go? Do you want the oxygen to stay in your lungs?
No, you want it to disperse throughout your entire body.
And how is it going to disperse through your entire body? Where does it have to go?
So then it goes to the cells, and the cells go throughout the body.
How does it go to the cells? What cells? So it goes to some sort of cells.
Blood cells?
Red
Yeah,
blood.
your red blood cells, that's your blood, right? Your red blood cells, the function of red blood cells is to carry oxygen around. So the function of your blood, the function of your cardiovascular system is to deliver oxygen and also other nutrients to everywhere in your body, right? The blood, the red blood cells never leave the arteries or the veins, right? So when you're here, we're talking about taking in from the lungs, you have a whole bunch of oxygen in the air sac here, right? And then you have blood vessels that are closely associated with it. How does the oxygen get from the lung to the blood, essentially, it's going to end up on red blood cells, right? Hemoglobin will carry that oxygen around. It's by diffusion. So it moves based on its concentration gradient. You have a high level of oxygen in the lungs, a lower level in the blood, and that's what drives that oxygen to move into the blood. It's just simple diffusion, okay? There is no complex mechanism. It's just that there's a lower concentration here, higher concentration here, and that's why it moves that way. It can diffuse across the membrane, and that's how it gets there. Then once the oxygen is on your red blood cells in your blood, right, it then moves around to all the rest of the parts of your body. Again, you'll have other capillary networks at all your other organs and tissues, right? And that oxygen will ultimately diffuse out of the blood vessels and then to the cells, to your tissue cells, okay? But the point I wanted to make here is that the movement of oxygen from the lungs to the blood is achieved by diffusion, right? And it happens because there's a high concentration here and a low concentration here. Molecules have the tendency to move from high concentration to low, and oxygen behaves that same way. Okay, does anyone have a question about that? All right, I'm basically going to end here. Again, if you didn't listen to this lecture, you need to. The concept of osmosis here and movement of things across the membrane is not that it's like super difficult, but it does take a little time to understand. So when you're talking about osmosis, what kind of diffusion? What are we talking about? Are we talking about the solute moving or what? What are we talking about for osmosis? What's the definition of osmosis?
The movement of water molecules.
Correct, the movement of water molecules. So, you know, we said that you can think of, you know, you have a solute and a solvent, right, in a solution. Thank you, Mace. I'm doing something. I'll be done in 2 seconds. There you go. And we were thinking about the movement of the solute, right? Our dye molecule was the thing that we were talking about. Well, when you talk about osmosis, you're talking about how is water moving, okay? So diffusion or osmosis is the diffusion of water, the movement of water from high concentration to low, okay? So like I said, make sure you really, you know, spend some time, you know, specifically with this part. I mean, I kind of just went through the whole first part of the lecture, but there's a lot more to say, okay? And the concept of osmosis is super important to biology, okay? And again, we do go over this in our diffusion and osmosis lab too. So it's something that we hit on a couple of times, but it is a very important concept, right? Particularly, we should know what we mean by isotonic and hypotonic and hypertonic, okay? So again, if it's a little confusing kind of when you first hear it, all right, it's okay. We are going to talk about it multiple times, okay? All right, so I'm going to end there. Does anyone have any questions before we go? And if you have individual questions, I will stay on as well. But lab will start at 3 o'clock. If you're not coming in person, sorry about my dog barking, you can log in to the lab meeting at 3 o'clock. Otherwise, I'll see you guys in person at 3, okay? All right, guys. So if you don't have any other questions, then you're free to go, and I'll see everyone at 3 who's in lab today.
Thank you.
Professor, can I ask a question?
Thank you.
Okay. So relating to our lab will be at 4:30, right?
Yeah. Are you going to be out of class like 4:30, you think?
We'll be out of class by 4:15.
Okay, so come, if you can, just come as soon as you're done with class. Is that
Okay.
Yeah, well, I don't know who else is planning on coming in person, but yeah, just come as soon as you're done, okay?
Okay. Thank you.
Yep. Oh my goodness. Other questions, guys? Come on, Paul.
Did you receive my I didn't send it to you, but I sent it to the college, and I haven't heard anything back from them. I sent a remote request to go fully remote for the remainder of the semester.
Yeah, I have no idea. I don't see any of that. I mean, for me, it doesn't matter as long as you're here. If you're staying remote, that's fine.
Yeah, I'll be completely remote. I was just wondering how that works with like, because you had assigned us lab partners and stuff like that, and like we had the lab report, which had like to be like done in person.
I didn't assign you guys lab partners. And all the labs you can join via Zoom, there's an assignment that you're going to be doing via Zoom. I mean, the only thing I scheduled out was when people were coming so that I didn't have too many people in the lab at once.
Oh, okay. I guess I got confused and I thought the names on that list were
No.
like partners. Okay.
Now we're not even doing that anyway. Everyone's going to be coming to lab at 3:00. Okay. Whether it's Zoom or in person.
So I just like whenever like I have like lab reports, I just write whatever like answers you guys get in the lab.
Yeah, you're going to follow along with what we're doing. I mean, you still have the other labs that are assigned on Moodle, right? That's separate, but the in-person labs, yeah, you're going to take, so I guess you came into class late. I went over this.
Yeah, I was having problems getting into the website.
So for instance, for this lab, for today, right? You guys that are on Zoom, there's an assignment on Moodle. You're just going to click it and there's a Word document. This is the Word document that everyone's going to be filling out. Okay.
Yeah.
So you guys that are on Zoom, I'll just be following along and filling it out along with the people that are doing the lab. You'll submit it via Moodle and they're just going to give me a hard copy of it, but it's the same exact thing. Okay. So there, like I said, on Moodle, there's an assignment that says in-person lab. It's in this week's module.
Yeah, I saw that. Okay.
Have that and you're just going to be completing it along with everyone else as they do it.
Does anything change in terms of like grading, or is there anything I should worry about?
I mean, the only thing that changes is that those people that come to in-person lab, they just get a slight advantage in that I'll end up dropping one of their lab practical grades, and you guys that don't come in person won't have that opportunity. But honestly, it's not really gonna, it doesn't really affect you guys negatively. It just gives them a slight advantage or a bonus for coming in person. So there's nothing you really need to worry about.
Okay. And I was wondering, whenever you get a chance, could we have like a private meeting?
Sure. What do you, I mean, probably next week, unless it's something that's like super pertinent. Wait, you know what? I gotta stop recording.
Because you're my advisor as well.